everyone. Thank you for joining on this Saturday from your busy schedule. पाकिस्तान से भी लोगों को खुशामदीद और यहाँ से भी जो लोग ज्वाइन कर रहे हैं बहुत शुक्रिया आज हमारा बहुत it's a unique topic which is not very well known, at least even me myself. I'll be very keenly listening to this because genetics, especially in Pakistan, what is going on, what is the status, what are the opportunities for the diaspora, Pakistani diaspora, which is the main target audience here, whether they are in UK, US, America, Australia, or wherever they are, and would like to join Pakistani colleagues like Aisha and Jawad, who will be speaking. Um, it's a great opportunity to learn where are the gaps or challenges or what exciting things they have already accomplished. A lot of time we think that things are not moving in Pakistan or things are not, at a level where we can go in and participate. But I think we are getting surprised every week, every Saturday when we started this webinar series, how many local Pakistani researchers and entrepreneurs and organizers have done such tremendous job that was very exciting at the same time and inspiring also. The idea of this whole series is that we as we are exploring the landscape of research and innovation in healthcare, and as we are learning, maybe some of us or many of us will be excited enough to go and participate and collaborate, as well as maybe if there are, innovative technologies which need some investment or collaboration from the companies here, we may also do that. Because as a reminder to everyone, Merit Committee has the last T in it as transfer of technology. So Merit is medical research. This is the medical education, research, international training, and transfer of technology. That's what Merit stands for. So with that, I will invite our speakers for today, Muhammad Jawad Hassan. Dr. Jawad Hassan is PhD and Associate Professor of Human Genetics in Department of Biological Sciences in National University of Medical Sciences in Pakistan. And in the panel, we have Dr. Aisha Moyadin, who we know have done such a great job in NUMS, because Merit has been working with NUMS for a while now. So please, Professor Jawad, you go ahead. And if you please tell us your background and then start your presentation.
Thank you very much, Professor Shahid and Danish Bhatti Saab, and all the members and all the colleagues for Association of Pakistani Physicians of North America, and of course the medical education, research, international training, and transfer of technology Merit. I must say thanks and welcome to all the panelists, Professor Aisha Moyadin, Professor Dr. Tipu Siddiq, and Professor Saqib Mahmood, who are there, and all the people who are listening to me. So topic for today is really unique for me as well, of course, because genetics research in Pakistan is usually, as you will see in the slides, is something new to our clinical friends in Pakistan. Whenever we talk, they say, okay, it's fine, but then there are some problems. I will, inshallah ta'ala, discuss in the slide. You already introduced me. Thank you very much. So the topic is genetic research and its translation to clinical trials in Pakistan. So I think without any further delay, I will move to my slide. And this is as per norms, this is the declaration.
Dr. Jawad, if you want to share your screen, that'll be great.
I think I've shared it already. I will. I try to do it again. I think I've shared it.
Please, thank you.
Solid share. Okay, and now
Got
let
it.
me
Yep.
know.
We can
Share
see.
now. Okay, okay. So, so as per the norms, this is the slide for the declaration of any competing interest, and there is no competing interest for this presentation. And now I will just give you the glimpse. I will follow this outline. We will discuss identification of the disease-associated genes, what methods used globally, and of course in Pakistan, and how we actually identify the gene mutations. And then after this, the translational impact of this identification of genes. What are the translation impacts? Why we do this thing? And then we will move towards some of the drugs which are approved and some of the clinical trials which are ongoing for the genetic diseases globally. And after this background, which is the global background, then we will move to Pakistani scenario. What's going on in the area of the genetic disease research? What are the issues? What are the problems? And then, of course, as a researcher, we will try to move towards some way forward. Then, of course, there are problems, and what we can do to solve those problems. So I will follow this brief outline. Move to this about the rare disease. This has been taken from the Journal of Community Genetics recently, and those regions, they are the World Health Organization regions, EMR, Africans and Southeast Asian regions, and Europe and America and the world and West Europe. This is just to show that, of course, this is a rare disease, the genetic disease, but there are issues and we cannot ignore it at all, that because this is the rare disease, so they are too rare, so there is no need to research on it, or there is no need to invest on it. Now, this is rare, but there are socioeconomic problems associated with this, so we have to work on it. This is the issues. And if you can see this East Mediterranean regions, Africa and Southeast Asian regions, the consanguinity, the marriages within the families, they are actually one of the major causes of spread of this disease in the families and in the population. So we have autosomal recessive or autosomal dominant or X-linked or Y-linked, as we know in the classical way the monogenic disease presents. So they are the forms, and because to erase polymerase, the polymerase adds the mutation somehow in the DNA, and then they inherit it in different forms into the next generations. And when we marry within the family, so the genome is of course accumulated, and the homozygosity increases in the next generations. So we have the diseases, especially the recessive. If we move on, and now they are the processes by which we identify the genes. How we identify the disease-associated genes. It starts with identification and enrollment of families. If there are multiple affected individuals in a family, we have to identify them first and then to enroll them in a research project. As we are in the universities, so we enroll them in the research project. In the hospital, of course, family didn't come. One individual usually came, because these guys usually they are from the poor socioeconomic status. So even if there are multiple affected individuals in the family, only one or maybe two will reach to a tertiary care hospital in neurology or some other field. And now, if you recruit from hospitals, you will find a rare number of diseases. Usually what we do, we go into the field, we go into the towns and into the areas where our students, they have the information that of course we have in our area affected families, in our area affected families. Then we go there and we identify the affected individuals for a genetic disease. It's hearing impairment, it's epilepsy, it's some sort of skeletal disorder, it is some sort of neurodevelopmental disorder. We go there and then we recruit them, and we identify them clinically, of course. But you know that in the field, you cannot identify all the things. So at times you have to move them from their town, which is far away usually, to a hospital nearby or to some tertiary care hospital in some big city. This is not the easy task. But of course, the researchers doing in Pakistan, in isolation usually in different universities, they're trying to do this in collaboration with the hospital and with their colleagues working in the hospital. Okay, so once you have this family enrollment, then of course you draw the pedigree, which is important, and the clinical evaluations performed. Next, we move to genetic testing. Now, there are strategies to do the genetic testing. If there is one affected individual, then of course we cannot do for homozygosity mapping. This technique is extensively used in past 20 years, especially where you have multiple affected individuals in the families, and you try to locate the locus, the regions of the marker allele homozygosity is shared among all the affected individuals. And then if you excluded first the known loci of the disease or the phenotype is known, then you will move towards the genome-wide homozygosity. It means that the already reported genes are not mutated in that particular family. There might be some new gene there. So, but if there is only one individual in the family, the statistical score would not be that much available. So we will go for candidate gene search. But for this, we have to have the accurate clinical picture. If we are sure what is the disease, only then we can go to candidate gene search. Without that, it is not possible. And then we do the Sanger sequencing, of course. If we know that, let's suppose there is congenital pain insensitivity, this is a disease, and we have the gene SCN9A for this, let's suppose. So if this is the disease and one of the strong candidate genes is SCN9A, then we can directly sequence that gene. So this is what the candidate gene search means. But if we had moved to the genome-wide homozygosity mapping, then of course we have a region of homozygosity. The locus, it might be 25 MB, or it might be 2 MB, it might be 2 centimorgon, or it might be 12 centimorgon on a chromosome. The classical words used for this. And for this, classically, we do Sanger sequencing of the strong candidate genes present within that particular locus. And now we have exome sequencing for us as well, the next generation sequencing. I will elaborate this in detail. The whole exome sequencing details are there, and we will focus on the variants which are present within the locus. So finally, we will reach to the pathogenic variant. But of course, for segregation analysis in a family, that exactly if this is autosomal recessive, then the parents are heterozygous for the mutations, and all the affected are homozygous, then we have to do the Sanger sequencing again. After all this, there is a variant detection. Finally, we are able to detect the variant. Then there are two ways to confirm. One, of course, is the segregation within the family. And along with this, we have the bioinformatics validation. Now we have so many tools in silico tools to predict whether the variant we are saying is pathogenic is really pathogenic or not. That variant is disease causing or not. Then we use the bioinformatic validation. And towards the end, of course, the final validation is the functional characterization of that variant. You have to have some sort of animal model. And in that animal model, whether it's mice or zebrafish or Drosophila or any one other, you will create that mutation, and then you will see, is this the phenotype, cellular phenotype is similar or not? So this is actually the way in which we try to identify the disease associated variants. If we talk about especially to Pakistani scenario, in Pakistan, we have multiple affected families. We marry usually within the families. So homozygosity mapping, even today, is the best way to detect the locus in Pakistani families, especially for the new ones, I believe. But usually we combine the techniques. We use the homozygosity mapping. And then in Pakistan, of course, we don't have the NGS. So we collaborate with the people in the universities in America, in Europe, in Australia, or nowadays in China as well. And then we do the NGS there and then combine the data in the collaboration. So they are the techniques. Classically, I just do the history, restriction, fragment length, polymorphism used in 80s, then the microsatellite markers, 400 to 500 markers usually we used. They were used in 90s and up to 2010. After that, we have the single nucleotide polymorphism, SNP markers, which are, of course, stable and they have the dense coverage. They are present across the genome and then they are fluorescently labeled. And we have the softwares to interpret the genotyping results, but exactly what the affected individual is homozygous for a certain region of genome. And that certain region is shared among all the affected individuals. So this is the locus. Locus means the region that contains the gene which is mutated and going to cause the disease in the family. So we finally reach there by using these techniques. I'm not sure whether everyone is really understanding this, but I'm trying to make it as easy as possible for everyone. So this is one of the ways, the linkage analysis and the Lord score and homozygosity mapping. This is the way in Pakistan. I still believe that we have multiple affected individuals in the family. So we should use this technique before going to the NGS, because that is costly. And of course, our patients, they cannot bear those. And in the research, we don't have enough funding for the higher techniques. So we need to be careful by choosing which technique we are going to choose for resolving a particular family. So now if we move onwards, we all know this is the Sanger sequencing, and we can do this for validation or for a specific gene. If we know, we just sequence that gene to know which mutation is there and which family. And they are the pathways and they are the standard techniques we use, and in the labs and in the pathology labs and in the research labs, Sanger is there since long and it's still effective. If we move on, then the NGS workflows are there. The next generation sequencing, this is different from the classical Sanger sequencing. We chew up the DNA into fragments, then we adapt the molecules bound to DNA, the adapters, and then we put it up. We made the libraries, then they go into the equipment, and equipment has a software. Then exactly when the data came out, the mapping reads, the visualizations, and of course, finally, we know what is the final sequence with us. And then we compare it with the standard sequences available of the human genome. So this is what all this NGS goes on. If you see what are the different types in the lower left panel, the mostly used is the targeted resequencing, which is related to the genetic disease. Either this is the whole exome sequencing or the targeted panel sequencing. This targeted panel sequencing is clinically the most important thing, especially in the families where we don't have enough family members to start with homozygosity mapping. The theory is that they have created the panels, like this a smaller exome sequencing, you can say that they created the panels of the genes for a particular disease, those genes which are already reported. Let's say in epilepsy, many companies are available globally, and they have created the panel of genes for genetic epilepsy, up to 200 or 250 genes in a panel. And for particular affected individuals, we just sequence that panel in that individual, and we reach the conclusion. If out of those 250 genes, the mutation is there, it's fine, it is resolved. But if not there, then it means that there is something new in it, something novel. Then you can go to whole exome or to some whole genome sequencing. And if just a historical perspective on the lower right, you can see that with the advent of this next generation sequencing, the KBs of DNA per day per machine increased tremendously. So this NGS actually, it changed the whole scene and the identification of the mutations or resolve of the family. So then we move here, we have the in silico validation, the pathogenic variants. We have different softwares. SIFT, polyphen, CAD, mutation taster. They actually predict based on different algorithms that is the mutation is pathogenic, if the proline to alanine change is really pathogenic or not. And on the other side, they have the data like 1000 genome project or some other genome projects. The data is there and that contains the population data from different global populations. So we compare the frequency of a particular mutation. If that mutation or that variant rather is available in the control somewhere, then it shouldn't be considered as a mutation, but this is available in the normal individual in these databases. So this is one of the ways to predict whether these pathogenic variants are, whether the variant identify it is pathogenic or not. This is one of the ways of doing this. Then the other way of validation, of course, is when you move inside the cell, or it's in vivo in vitro validation. And in this article, the molecular aspect of medicine, this is very clearly elaborated. I will try to elaborate in a small way where it depends which type of genes you are going to test. In the upper panel, if you see, they actually test the hereditary CRC gene variants. And in this panel, if these type of genes are there which are involved in particular pathways, like in say NMR activity, tolerance to methylation agents, then which type of model we will choose to check whether our variant is pathogenic or not. Then we may choose cell-free assays, or maybe E. coli, or then we use yeast, or the mammalian cell, or then even the pluripotent stem cells, the IPCs, which are the future strategies. And for RNA studies, we can use RT-PCR and sequencing, or the qRT-PCR, or for other protein studies, we have to have the protein-protein interactions, or the protein-DNA binding interactions, and the protein expression stability, just to show you that there are the ways, and there might be many others, to validate that which is your mutation is really pathogenic or not. You have in silico, you can check the things on bioinformatics, then you can check the things in the cells that our mutation is really a pathogenic variant. I'm going through this because only after this you can go or you can think about some sort of the gene alterations in the mouse model, and then you can go for a trial into some sort of a drug to alter that in the patient. So it is really important to go through all these mechanisms. So moving on very briefly, now we move to gene therapy. This is the timeline. We have the traditional gene therapy in 80s and 90s. And then this name is the CRISPR-Cas9, which is a bacterial enzyme system. And we actually identify this. Bacteria use this for their immunity. And we identify this for ourselves to change the DNA sequence where we want, and then to put that transgene into either directly into the cells or organs, or to some others, let's say in the stem cells, and then put those stem cells into the organs. Two ways are there. So this is the timeline. You can see from 1999, one of the patients died for these experiments as well. This is important to show, and this is one of the problems where the scientists are not able to find out so many answers to the problems in genetic disease, because the experiments, they cost even human life. So we have to be careful. And if you see towards the end, and from 2017, the CRISPR clinical trials are in process, 2 in China and 2 in USA for a different type of disorder. And they started from the traditional ways, and then now the CRISPR gene therapy is there. The people initially used the mouse models, and now the zebrafish is there to use, and people use the zebrafish model. They say it's too easy. Some people believe that the mouse model are more easy. And again, depends which is your disease and which is the gene actually you're going to alter, you're going to change in the patient. If you move on, just as I told before, 2 ways. We have either we made the transgene, we changed the genic sequence which is mutated to the normal, and then we either we put directly into the organ into the affected individual, or the cell-based delivery is there, that we put that transgene into some cells and then put those cells after certain modifications into the organ. So two ways are there. I'm not talking at the moment about the transgene, how we made the transgene. We have the viral vectors usually, and then maybe adeno-associated viral vectors, lentiviral vectors. Then we have the liposomes. We have the bacterial plasmids. We put the gene into them, and then finally it moves into the organ where we want to put that gene. And now, moving towards the trial, and this is the global picture so far. Usually it has been said very rightly in this article of translational medicine, and they said that this is the value of death. And the value of death is between the translation from basic science to human studies, from preclinical to clinical. Here is the value of death. And here are the problems which are faced by the scientists globally. If we want to start, or if we are interested in gene therapy trials of genetic diseases in Pakistan, then we must know about this value of death. This value of death exists even in USA. So we need to be very much careful about our resources, about everything which we have, what we have to add, what in types of, let's say, HR, or the training of the people, or the infrastructure, or all other things, ethics, ethical approvals, the legal approvals, everything. So we need to have a look on all these processes, of all those scientific processes, and the legal and the ethical processes there. And so when you put something in mice, you find very good results. Okay, it's very good. We have this drug with us. It can change this DNA, and if you change the DNA of a particular gene in the mice, it is okay. But when you have this drug and you come to the phase 0, or phase 1, or phase 2 trials, the drugs usually they fail. And the people say that half of the drugs which are there, they fail even reaching to the phase 3 trials. One of the issues is might be the difference in the gene expression in the mice and the human. Someone has told that the two animals, they were talking to each other, that have you been vaccinated with the COVID? And the one said, no, they are testing on human, and after that I've been vaccinated. So actually, if all of us are mice, it's quite okay. Most of the diseases have been cured. But we are not mice, we are human. So in the mice, it's easy to cure, but there is lots of changes in the gene expression. The gene is same, but the expression patterns are quite different. So this is one of the reasons that the things fail. And we need to learn about these things if you want to start. So moving on, there are some of the approved gene therapy drugs. It's about 20 till 2019 in an article review. It might be 3 or 4 added to it, I don't know. And they have both in vivo gene therapy and ex vivo gene therapy products so far for different companies, different countries, and they are for, of course, different disorders. You can see this thing, the gene TP53 VGF, retinitis, pre-mRNA, SMN2, the spinal muscular atrophy. Then we have adenovirus here in this gene. That's exon 51 of DMD, the Duchenne-Becker muscular dystrophy. They want to correct this mutation. Then on the right side, we have the TGF-beta and the CD19, CD137, ADA gene. We have the genes, we have the disorders, and these drugs, they are approved drugs. But again, not all of these approved drugs, they reach the clinic. They don't reach the clinic because of many issues. So we have to be too much careful. And Pakistan, of course, the genetics study is there, and we have so many publications from Pakistan and Pakistani families. But to move to this trials, we have to have too many things in it, and we will discuss in next slide. So they are, you can see from the USA, the approved drugs, FDA approved, and from the Europe, we have this one from the China, South Korea, only one, then the Russia. And whole of this Africa and the Middle East and South Asia, they are white. No drug so far in the genetic disease. Moving on, there are some of the products which are under trial, and the details are there. I'm not going to read it. The details are there. Anyone can see these slides. I hope they are available to the people. So anyone can see that these are the drugs for gene therapy, and they are till 2019. It might be possible some of them they have been approved, but I don't have detail. Sorry for this. You can add 5 or 6 to that, 20 from this, just on average. They are the slides say they are about 28 in total, which are in the clinical trial. Okay. Now, from this onwards, the actual topic is I will start, not more slides. Here I am. The genetics research in Pakistan, there are a glimpse. I've shown you the research papers, or I will try it in one slide, the research paper from different groups. They are working in Pakistan. I will show you. So Pakistan is a big population, and we marry within the families. We have consanguinity rates high in Pakistan. Many studies they have reported from 40 to up to 62% marriages in close families. So with this, of course, there is a chance of the autosomal recessive disorders. Most of the disorders which are prevalent, although we don't have the data like this, like we have shown in the very first slide, but from this online data, you can see the hearing impairment, congenital hearing impairment might be the most common genetic disease. Then we have in Pakistan, of course, this is from the published data. It's not the epidemiology. I'm just telling you the neurological disorders, including mental retardation, microcephaly syndrome, and some other epilepsy. They are there. Then we have the skeletal and neurodevelopmental disorders. Then we have the skin disorders, of course. Yeah. And then, of course, towards the end, the eye disorders, retinitis pigmentosa, Usher syndrome or Usher syndrome, what you can say, they are there in Pakistani families. And with these disorders, families have been reported from Pakistan. Many genes of these disorders firstly reported in Pakistani families. Microcephaly genes first time reported in Pakistani families. So many hearing impairment genes, more than 20 out of 100 so far reported. They have been reported from Pakistani families first time. Of course, that they are reported in the other families globally, but first time they were reported from Pakistani families. Most of these studies, they have been done in collaboration with some labs outside Pakistan. There are groups in Pakistan. If I go to show you the history, I think the KRL lab started it firstly, and Professor Dr. Qasim Mehdi and his group, they published first locus, DFNB8 for hearing impairment. Then, of course, the other papers. Then we have in Lahore a very good group of Sheikh Riazuddin Saab in Center of Excellence in Molecular Biology, and they published very good papers for hearing impairment and the genes identification and the mutation spectrum. Then we have the group in Quaid-e-Azam University, Professor Wasim Ahmed, and their students, where I did my PhD. And usually the skin disorders published there, from there, along with the microcephaly and hearing impairment and skeletal disorder. Then we have the group working in Faisalabad in National Institute of Biotechnology and Genetic Engineering, Professor Shahid Beg, and of course they also published tremendous articles in collaboration with their collaborators. Then we have the people working on genetic disorders in Balochistan, in University of Balochistan and in UTEMS, Dr. Ayub Kakad, Dr. Wali Tareen. Then we have the people in Peshawar. They are working on genetic disorders in Islamia College. They have a genomic center and they are working. So people are working in Pakistan, but from universities they are working. Unfortunately, from clinical side, I didn't know anyone. If there is someone, that is my mistake, from the clinical side who recruit the families and publish them these gene mutation studies in Pakistan or in collaboration with a foreign. All of these studies have been published from the university, Quaid-e-Azam University or University of Health Sciences Lahore or some KRL lab or some. So the clinicians' involvement is too less in this, and I think we need to address this issue if we want to increase this genetics studies in Pakistan, and if we want to go towards the gene therapy for these disorders in Pakistan. We have to involve the clinicians. Rather, clinicians have to involve the basic scientists in it. It's other way around. But this is the area where I think we need to think. If I move more on what they are my points. Reasons for the success of genetic studies. Genetic studies are really done in Pakistan, but not after this. The expression studies or the gene therapy trials or some other thing are not there. Up till genetic studies, people are working, and of course, they have large inbred families, so we have the option to work on them. Scientifically speaking, it's easy to handle with DNA. It's really difficult to handle RNA or the expression studies or the animal models or the type thing. You need more money, you need more infrastructure for this. And the handling of DNA is quite, it's a stable molecule. Third, and the most important thing is that the Pakistani scientists, they have effective collaborations. And almost all of these papers in which the gene is identified first time, they are identified through a good collaboration outside Pakistan. And in the last lines, I just go through the PubMed. And for this, one of my colleagues at NUMS really helped me a lot. If you go type these words in the PubMed, you have these findings just to show that the Pakistani scientists are working on the genetic studies in Pakistan. In Pakistan, in collaboration or independently. Now, the mutation detection is done independently, but identification of the genes and the in vivo or in vitro studies to validate the mutations, they are rarely done inside Pakistan and in collaboration with the labs outside Pakistan. Moving on, you see this thing. Whenever either linkage analysis or gene expression, whole exome sequencing, here NGS means other than whole exome sequencing, the targeted panel or the type list. GWAS, the genome-wide association studies for the complex diseases, I didn't include the data. Here I just include monogenic. Then the knockout mice studies, and towards the end are the clinical trial or gene therapy. So there is no studies. Here we have absolute zero. Onwards, you can see that linkage analysis, we have 165 publications solely in Pakistan, no foreign authors. So we can do this thing in Pakistan. In Pakistan, linkage analysis studies to map the locus, yes, we can map. If the gene is known, yes, even we can sequence the mutation, we can publish the mutation. But if you move on towards the complex things, we do not have any functional whole exome or NGS platform in Pakistan right now, as per best of my knowledge. And then the knockout mice or the clinical trials, even no need to think about it at the moment. So there are some of the studies in collaboration with some foreign labs. But if you want to see the data exclusively done inside Pakistan, no, we didn't do exclusively in Pakistan. We need collaboration, our infrastructure, our funding, and of course, our ability to do this, our experience to do these things, this is lacking. Moving towards the more thing. So the last two slides, and they are actually the slides I think we need to discuss. But before reaching this, we have to go on through a pathway. There are some issues. We want to do the gene therapy trials in Pakistan. So some of the issues are the scientific challenges, which are global, we discussed earlier. There are gene expression pattern differences, human versus mice, the value of depth is there. Then the ethical approvals is not easy anywhere, especially if you are in USA. I know some of the colleagues are working in USA. They're trying to have a gene therapy protocol for Lafora epilepsy, a type of epilepsy, and now they have moved to Spain for this because they are unable to get ethical approval. So this is the global challenge. This challenge is in Pakistan as well. Number two is the understanding. We need to think that inside Pakistan, less people are there who really understand the science of clinical trials, and of course this genetic disease and the hardcore experimental science involved in it. And of course, there is lacking of the understanding of the procedures of the clinical trials, ethical and legal both. I am a member of IRB in a tertiary care hospital in Islamabad. And I know that there are very, very few people who really know what these clinical trial statistics and ethics means exactly. So if they are there to approve these things, even in the regulatory bodies, then I think there will be problems. So we have to address these things. They are the challenges. I don't have other word in this three than negligible. I want to write something more than this, but it's not available. The industry in Pakistan, especially the pharma industry, mostly they are packing or repacking the compounds already under some license from some external pharma. So they are not interested in research at all. And if an assistant professor from a university went there to collaborate, I tell you, the security guard will let him in. As well as these type of things are there. So there is role of industry is there. We need to make them include in this process. Funding and infrastructure is again a global process, but here I must say that from past many years, rather decades, Pakistan never exceed spending more than 0.3% of its GDP on research and development. Period. This is far less than the SAARC countries, where most of the countries spend more than 15%. So here is the issue. This overall for research and development, and of course for this type of gene therapy and genetic next generation sequencing and mouse models, you need the infrastructure and you need the funding, which is not available. The bureaucracy and red tape issues, they are there globally, they are there, and in Pakistan, we know they are there. Now, towards the last point, the clinical scientists in Pakistan, in my opinion, they have less or no protected time for research. Most of our clinicians in our private hospitals, the hospital administration is not really interested in it. That dear people who are actually checking the patient, they should spare some time and do some research. No, they are not interested in it. I'm too much straightforward, but this is the reality. And one other thing is, which will go in the last slide, which is the next, then there is a lack of motivation I see. One of my colleague, which was the former president of the Pakistan Society of Neurology, he told me that about 200 to 250 practicing neurologists are in Pakistan. And what big population we have. So if we ask 20 or 25 of them to please spare time on weekly basis for research, and we walk to deliberately, what would be the fate of the patients? So this is the issue. We need to solve this. The last one slide, the way forward. There are issues, what we should do. The collaboration and the teamwork is one thing that we should do. And clinician and scientists inside Pakistan, the clinician in the hospital and the biologist in the university, they should collaborate. Until and unless they collaborate, we cannot move ahead. And the second thing is that the institutions, they should contact the industry, and then the people from the both sides, the professors or the researchers, they should contact each other. If you send your researcher to industry, I don't think so the things will move. Hiring of experts and the training of the existing people. In Pakistan, as per best of my knowledge, I don't know a single person who is really involved in the gene therapy clinical trials in America or in Europe, and now he is working in Pakistan. I don't know a single person. So we need experts there. And of course, when you have the expert, you have to be at the cost of hiring. If you say कि जी वो हमने grade 17 में आदमी लेना है, तो this will not work. Then I think if you make a collaborative team of these two, then train these two, train the biology people in the clinical science or in the hospital work, not ये कि आप उन्हें MBBS में ले जाएं, but train them in some sort of clinical training. And those clinical scientists, please train them in the hardcore experimental sciences through degree programs, through certification. Only then we will have a good team, and then you will hire a good scientist expert about them and give them the task, and inshallah, then you can have the things. And this is the thing, the point number 3, I don't know that whether this will reach to the people who are involved or not. This is really important. In medical colleges, in undergraduate and postgraduate, both, And as a postgraduate training program in clinical sciences, genetics should be included. Uh, if you don't include this thing, then there is no career pathway for our clinicians to work in the genetics. So they are not interested, they are not motivated to work in genetic disease. And of course, that will lead to clinical trial. Then the thing is the coordination between the regulatory bodies. This is too important. One degree or one program, one body accept it, and the other body says, no, we didn't accept it. So we have to have these all on one page. And the last thing is, of course, we need to have this database of the genetic disease and the regional mutations. And if we have this thing, only then we'll be able to move towards the carrier screening, genetic counseling and testing, and then finally towards the gene therapy. We need our mutations, and those drugs would be required. And I haven't written here one more thing, and I will just say that the perception of the common people about genetic disease and about medicine at large needs to be changed in Pakistan. And this might be done by the social scientists within the medical universities, within the hospitals. This is required. Until the perception of the people change, you cannot do these things. So towards the conclusion, we know the last thing that only 20 so far till 2019, we have the drugs. And out of those 20 drugs, not all are translated into the clinic. So the valley of death is even after the approval. No clinical trials in process in Pakistan for any genetic disease. And from genetic disease, I mean the monogenic. If there is any, please tell me, maybe I'm wrong. The thing is that there is the potential. Pakistan has a heavy burden of genetic diseases. Many genes were first identified from Pakistani families, though the potential is there. But there are ifs and buts if you want to invest in this area in Pakistan, and we need to learn all these things, the 360-degree rather study, and then to go to this area. And I must say from this slide, thanks to APPNA and thanks to Merit, thanks to all my panelists, and thanks to all of the people listening to this one. And this, I think, this is something that since last 20 years from my PhD to now, I'm working in Pakistan, and this quote always helped me in working in Pakistan.
Thank you.
Yes, sir. Yes, sir. Thank you very much. They are my PhD students working with me, and here I am working with and sampling at different areas. My own region in Punjab, Jawad, in the Thal, where I was in my PhD, and this is in the tribal areas where I collected some of the samples just to show you. So thank you very much, everyone.
Thank you, Professor Jawad. It was really enlightening, very straightforward. And as we know that there are so many challenges, so many issues, but of course we have a very illustrious panel today. And so we can discuss some from Pakistan and hear how, if there's a pathway to move forward in this important discipline. So first and foremost, we are very honored that one of the most illustrious, not only here in US, but in the world of genetics, Professor Tipu Siddiqi is with us, and he has a very long, really introduction. So I would just highlight few things because it would be unfair not to mention them. He is the Les Turner ALS Foundation Herbert C. Wensky Professor, Department of Neurology, Cell and Development Biology and Pathology in Northwestern University Feinberg School of Medicine in Chicago, Illinois. He is the one who he was the first one who developed the neurogenetic lab in Northwestern in 1991. And since then, his, the work in ALS, the world knows about it. And recently it was noted that in the past 20 years, all the work that has been done in ALS world and the 10 seminal papers that came out, 5 of them came out of Dr. Siddiqi's lab. He has been consistently top doctors in the past 20 years in Chicago and Illinois area. is also honorary Doctor of Science from Lake Forest University of Lake Forest College in Illinois. From 2014 to 2020, he has been nominated as the 500 most influential Muslims in the world. And he also had has the president of the Iqbal Society in Chicago, Illinois. But for us, his major contribution is that he brought neurologists in America and North America together, and founder of Pakistan International Neuroscience Society, numerous contribution to develop neurology in Pakistan and here. So we always look forward to his advice, take inspiration from him. And today, I think because of this challenges and all that, I think he will be the best person to guide us and maybe set the stage for what questions and what pathway can be devised and set the stage for that for the rest of our panelists, who I'll introduce after we get the comments and from Professor Siddiqi.
So in the interest of time, I don't want to monopolize the stage. And to George Bernard Shaw, who was a womanizer, I just want to say what Iqbal said, "Apni duniya aap paida kar agar zindo mein hai." So this is a challenge that is for any poor country, and especially an ostracized country like Pakistan, where big pharma will not come to you, where major universities will not invest, where very large moneyed foundations will not invest. Otherwise, they would have done it. For all the reasons that Professor Jawad pointed out. And I really congratulate him for working in very difficult circumstances and producing original papers in identification of genes in Pakistan and recognizing the problem. Obviously, the first thing is to recognize the problem. And so I had the good fortune to be able to see some of these issues, and I worked with some of the most illustrious and dedicated Pakistani geneticists, including Wasim Ahmed. We just missed the microcephaly. We had to run all those blots. He had sent a very good technician to my lab, and we just missed it because of scientific colonialism. You know, you had Pakistani families in England, and they had more resources that published. So Dr. Wasim Ahmed was not able to publish the original paper on microcephaly. Similarly, we collaborated with his friend, Fayyazul Haq. Fayyazul Haq is now in Saudi Arabia. He and Dr. Wasim went from village to collect samples. They took loans from banks to be able to do that. So they are real pioneers who put their belief system in science on the line. And Fayyazul Haq was working on skeletal disorders. He worked with one of the most prominent skeletal geneticists in the United States. And he worked in my laboratory for a while on his project. He is very unusual. His father, his family is from Bangladesh, but his father chose to opt for Pakistan. So he's completely loyal to Pakistan, and we don't really value these people. So anyway, the point is that we cannot go the route of the wealthy nations. We have our own problems. We cannot become just followers, as most of the so-called great universities in Pakistan, you know, I don't want to name them, prominent one in Lahore, another one in Karachi, a medical school in Karachi. They're just, as in the terms of Iqbal, they're taqlidi. So if you just look at genetic therapy, if you take spinal muscular atrophy and you take the new drug using a adeno-associated virus vector, it's $2 million a shot. So the whole system is wrong. It's a system based upon corporate medicine, and you don't want to invite corporate medicine into America. It is seeping in because the model systems and I mentioned the famous medical school in Karachi and the hospital. That is what they're oriented towards. It's they have reduced American medicine to McDonaldization. And so that is likely to happen in Pakistan if we just blindly follow what others are doing. So our problem is huge, as was pointed out by Professor Jawad, in terms of consanguinity, endogamy, and such like things, but there are also great advantages in it. And so the answer does not lie in what people are following. And as a student of Iqbal, I can tell you that he also was an original thinker, and he knew the answer did not lie in what the Indian National Congress was trying to project. The answer for the Muslims of South Asia lay elsewhere, and he was able to propound it in a way that went against the winds of the time. And he was successful because his revolution went to Iran, it went to Central Asia, and now it is with the Palestinians. So we need to do some original thinking as to how to solve this problem. I have some ideas, but they may not be the best ideas. People who are on the ground there have to figure them out. In the long run, let's talk about the long run. In the long run, one has to start with education. And I think Professor Jawad pointed that out, that there is a paucity of knowledge about genetics. Even though, if you look at the life of the Prophet, there was an issue about who was related to whom. And I have presented that before. And they brought in a person who could look at physiognomy and say how people were related. And so there was a question about paternity. And the Prophet was really pleased, and he told his wife Aisha that, look, it has been settled now, that this issue of paternity has been resolved, because he was a peacemaker. He was not somebody who wanted strife in society. But the point I'm trying to make is that this effort of trying to understand genetics has been going on for a very long time in various ways and various tools that were available to people through the ages. Now we are in a position to understand it better. We don't have a complete picture of it, obviously. We have a distorted picture. Of course, it is going to bring very unhappy ideas about history, about understanding that consanguinity, I should rather say that there is no such thing as piety based upon genealogy, because after the 11th generation, there is virtually nothing left except the Y chromosome. So it's going to make people very, very uncomfortable about those who make claims about genealogical superiority. But that's a good thing, isn't it? That we are really what we are by what we do and what we believe in, rather than our ancestry. So it's going to prick the bubble of ancestry, but we should be ready to accept the truth. And that truth is going to, as I said, profound effects on our society, and I think in the long run, a good thing. I don't want to name names, but we can do that another time. So long run is we must start from grade school. We must have, and we have those tools to explain to people how inheritance takes place, how people, even in countries like Saudi Arabia, it started actually in Cyprus. The United Nations wanted to bring the Turkish and Greek populations together, so they set up a genetic institute. My mentor was very prominent in that activity. And so they trained people. They came to Duke when I was at Duke, and they trained there and went back, and they were very successful, because what was the problem? The problem was Mediterranean diseases, which is also in Pakistan, the issue of blood disorders, which are inherited, and when they're homozygous, they're a terrible toll on society, and they're widespread. And so they were able to institute genetic counseling and be able to tell people who are going to get married if they carried the gene. And this has happened now in Saudi Arabia, where for thalassemia, you have to show a certificate that you have been tested. This is not a new idea. People used to do it for syphilis and HIV and so forth. So the question is that where do we start? Obviously, there are short-term issues and long-term issues. The long-term issues have to do with with knowledge, and it starts to people to understand right from the beginning how things are made and how the physical world works and what is the important genetic contributions. And of course, we are still in the process of learning, so we really can't say we have the last word. But that's where we start. And then for a nationwide effort, we really need to set up an institute. How it's going to be funded is something to be thought about, but you don't want to sell your assets to foreigners. So it has to be an indigenous effort where you set up an institute for genomics. We don't want to talk about genetics anymore. We want to talk about genomics. And what would be the purpose? The purpose would be to look at endogamous populations. It would be important to look at the burden of disease, to train at the district level, or maybe even at the Tehsil level, genetic counselors and genetic testing issues, because genetic therapy is not going to work for many reasons. I mean, we are doing it, but we are doing it mostly as scientists. We just published on CRISPR-Cas9 correction in the genetic mouse that we made for ALS in 1994. But the issue is, if the technology is here, we can take that associated virus and we can put things in it, but we don't know the off-target effects and many other things. There is genetic therapy, which have been used for a long time, for example, in Wilson's disease, where we can get rid of the copper and make a big effect on the patient. But the gene therapy issue, so where is it going to work? It's going to work in diseases like Duchenne muscular dystrophy because 1/3 of patients are new mutations. It is going to, we have to investigate Down syndrome. But for monogenic, highly penetrant disorders, we have to prevent them, and they're preventable. And there is no reason really, by education, it's a slow process, but the opposite of which, expense. If Biogen is going to charge $2 million for one shot, nobody can afford it for one disease. So you can see that our answer does not lie where the world wants to take it, where capital wants to take it. It has to go where human needs can be fulfilled, where we can at the same process educate our populations, where we can give autonomy to people who want to get married, and to take into consideration what their children are going to be like. So we have to reduce the probability of making a mistake less and less, and reduce the burden of suffering. I have been watching these videos of this couple. I forget now what they do, but they go around helping people in Faisalabad or other places. And it's really poignant if you look at the children living in those circumstances where they are retarded or they have skeletal defects, and how can a society continue to tolerate it? And obviously, we don't have the money for genetic therapy, and it may not succeed, and it may have off. So we have to prevent it. And that's the long-term answer. That's the long-term answer in America. That's the long-term answer for research. Yes, we can continue to do research. I think it's important, but there has to be a national institute for genomics. There has to be training institutes everywhere for genetic counselors, and obviously for physicians and nurses. And these are not very expensive things if we have the will. I mean, is it not a major problem? Yes, it is, because it will also give benefits that we don't expect. For example, we could set up virology labs. We could do sequencing of sewage. We could do sequencing anywhere we wanted about any emerging threat in the population. We could make vaccines. We could do a zillion things once we have the basis established for genomics. And we can do plant genomics. We can do so many things. I think so the national talent has to be gathered. People like Dr. Jawad and Wasim Ahmed and others, they need to be brought together. And, I think the late Qasim Mehdi, I met him one time. He was brought in by Abdul Qadir Khan. And, he couldn't publish some of his work because he thought he will be killed because the issue of this genealogical sanctity people have. But it will go away. Knowledge, you see, people are always negotiating with knowledge and information and new things, and they come to terms with it eventually. So my plea would be to have a series of conferences to brainstorm how to move forward in the short run. Obviously, you need to train people and you need to know the technologies and you need to do research. That's all fine. I'm talking about the public health problem. What's the biggest problem? Thalassemia, right? Thalassemias need to be prevented in Pakistan. You just cannot fix so many people. They have so many different mutations, but you can prevent them. And other countries have done it. They're good examples. So start from the beginning. We should write children's books about genetics, how things work in biology, how plants work, how animals work, how humans work, and what are the diseases that are produced. We should produce good quality videos about the terrible consequences of genetic disease. But still we are left with the problems like Duchenne. 1/3, it's such a huge gene that 1/3 of there are always going to be new patients. We're left with issues of chromosomal disorders. So there's a lot of places to do research for epigenetics, for developmental biology, what happens in the grandmother's womb. So there are lots and lots of opportunities to try and understand the environmental interaction. And then, of course, there are late onset disorders which are not fully penetrant, like neurodegenerations, which have environmental effects. And the very huge issue which has escaped investigation by genetics is the issues of mental health and psychiatric disorders. They have absolutely produced nothing. I mean, now with 4 million people with GWAS done, there has been a very large number of rare variants that have been associated, but one is at a loss. So it's a huge challenge that new researchers should be investigating, and new neuroscientists need to be trained to mate the imaging, the developmental biology of the nervous system with genetics. So we need interdisciplinary effort. And I'll stop there because I don't want to hog all the time. I've already done. Sorry about that.
Thank you, Professor Tipu. We always, mesmerized by your talk and very good points. And we may come back to you. I think let's get around with the panelists on the table. I think next, Professor Aisha Moyadin, who is the Professor of Biological Sciences in National University of Medical Sciences. And we always very impressed by the NUMS ORIC and met Professor Aisha many times. And I think it's all coming after 3-4 years of our collaboration with NUMS. The research was the main thing that we were impressed how NUMS is leading in that. And of course, APPNA Merit and PINS will contribute in that. So Professor Aisha, your comments and any advice there?
Assalamu alaikum, everyone, and thank you, Dr. Jawad, for a very good presentation. I'd also like to thank APPNA Merit for inviting me. I think Professor Tipu has covered many of the points that I would have put across. I do think I was lucky enough to be one of Dr. Qasim Mehdi's PhD students. And I would like to say what I think is missing is we started doing linkage analysis and studies and gene identification somewhere in the mid-90s. And unfortunately, in Pakistan, we have not really moved beyond the DNA analysis, simply because it's just been easier for us to send all our samples outside and have them analyzed outside. There hasn't been much of an effort to build any kind of infrastructure towards functional genomics or working in animal models and so on. So I think one thing that is very important is that we in Pakistan need to focus on that. We have a lot of very, very good researchers. We have a lot of people who have done PhDs who are coming back having done amazing work, but they simply don't find the infrastructure to support their expertise in Pakistan. That is one thing I think we need to work on. Another thing I think is to recognize clinical genomics or clinical genetics as a medical specialty in Pakistan. I think one reason we haven't moved forward with it is because medicine it's simply not recognized as a medical specialty. Doctors don't know much about it. And I think one thing you'll see in Pakistan is that patients do listen to medical doctors. They have a lot of respect for the medical doctors. So a lot of this advice, if it were coming from their clinicians, I think it would have a bigger impact. So I do think that our collaboration with, for the scientists to get together with the clinicians, that is something that is also very important. I do think that our clinicians in Pakistan are seriously lacking in this area, and they need to be trained, or the only way to actually get people is to recognize it as a clinical specialty. And I think that is something that needs to be done. Baki, as Professor Tipu said, I think clinical trials is something in Pakistan, in gene therapy and so on, is something way in the future. And we really need to focus on the preventative side of just getting people to understand what it is and the risks of marrying cousins, especially in families which already have a heavy disease burden. Unfortunately, I mean, I think all of us who've been collecting families with genetic diseases, even though they have several affected members in their families, they still would rather marry a cousin than somebody outside their families. This is something also which can only be corrected through education, as Professor Tipu said. So I think that is something that we all should collectively be working on. I think that's about it for me at the moment. Thank you.
Thank you, Professor Aisha. Next is Professor Saqib Mahmood, who's Associate Professor in Human Genetics and Molecular Biology in University of Health Sciences, Lahore. Saqib, Professor Saqib, please give your comments.
Assalamu alaikum, and thank you very much. First of all, Jawad, for an excellent presentation, and then to APPNA Merit for giving us, the geneticists as such, this opportunity to talk on this very important aspect of medicine. At least I'll be representing medical community as such, because I'm one of the few people who, after doing medicine, have gone into the genetics. And I believe, as Jawad told, Pakistan is considered to be a gold mine for genetic research as such, because we have large families, we have consanguineous marriages, and so a lot of data is being produced from Pakistan on genetic disorders. But unfortunately, that has not been translated back to the patient care. That's my main point as such, that it has not been translated back to the patient care. We don't have labs, diagnostic. There's no comprehensive diagnostic lab for genetics. And see, there's no use producing genetic counselors, or there's no use producing clinical genetics until and unless that is backed up by a comprehensive genetic lab. There are very patchy genetic tests being done, some in Aga Khan, some in, I believe, Armed Forces Institute of Pathology in Rawalpindi. But then the first thing we need is a comprehensive genetic diagnostic lab. And based on that diagnostic lab, we can work on the prevention, we can work on genetic counseling, we can work on the genetic research and all those things. And then the thing that is happening is a lot of blood is being sent abroad. As said by Dr. Aisha, what we are doing is, and as said by Jawad also, what we are doing is sending our samples abroad, and the data is being gathered abroad as such. We don't have a national genetic database. We need to have a national genetic database, and that is essential for genetic to progress in Pakistan. Talking about prevention, yes, of course, as Dr. Tipu said, I would like to mention here that we have a very robust thalassemia prevention program in Punjab now, and it's been running for the last 2-3 years, and it's spread all around Pakistan. So we are. It's not very gloomy as such. We are progressing slowly and steadily towards prevention programs, and we have to expand to other diseases also. Regarding clinicians being not interested in research, yes, it is true, but then there is a lot of research being done, especially in genetic disorders in Children's Hospital, Lahore. If you go into Dr. Huma Chima, she's a pediatric gastroenterologist. She has published quite a lot of papers on this inborn errors of metabolism. Then again, it's in collaboration with international collaborators. So nothing been done in indigenously. And hopefully we are trying to establish a comprehensive lab that will be announcing that in 4 to 5 months as such, a comprehensive diagnostic lab as such. And talking about genetic counselor and clinical geneticists, yes, we need support from APPNA that we should introduce these courses in Pakistan, produce clinicians who are into clinical genetics. They are pediatrician. They want to subspecialize in clinical genetics as such, and this is high time that these opportunities should be available as such, because we don't have any clinical genetics to train our local people. So we would be asking you people if you can help us out, and UHS would be very much interested. University of Health Sciences would be very much into collaborating with you people for these masters or diploma or certificate courses and all those things. And also about UHS is very much involved in clinical trials these days. We did rather, China is much into clinical trials in Pakistan these days, and University of Health Sciences has recently concluded a clinical trial for COVID vaccine, CONCINA, and now there's another trial for three-dose trial of China that is coming up in University of Health Sciences that has rather started, and other trials are also being in progress as such, but these are mostly for COVID as such. So regarding clinical trials, we don't have so many recognized clinical trial centers as such, nor the facilities. So it will take some time to establish those facilities and go into the clinical trials, especially for the genetic disorders, because we have a lot of metabolic disorders. There has to be enzyme replacement therapies. As Dr. Tibu said, it's very, very expensive. They are kind of orphan drugs that cannot be afforded even in the developed country. So how can we do that in Pakistan? So prevention is the best thing. So by the gist of my comments would be that we need to establish a good lab. We need clinicians, and clinicians are now interested in genetics. I've seen a lot of clinicians, they are asking for different tests. They are asking for their patients. They want to go for the diagnosis, and even you see our Rabban's pathology that has gone into molecular. So even medical students, they are not aware about genetic disorders and molecular pathology. So slowly and steadily, it's gaining ground in Pakistan, but it is taking some time. So I'm sure we'll be able to do something about that.
Thank you, Professor Saqib. In the interest of time, I would request participants to hang out some more, because the topic is so much of immense importance, and we have illustrious panel. And so, and also I request Danish to please collect some questions for the panelists after we get the panelist comments. Next, I will introduce our new member, Professor Tariq Zaman. He's a research professor at Michigan State University and have done research in Korea, Canada, and USA. His interest is in epilepsy. So Tariq Zaman, please tell us more about you and your vision, or how this collaboration with Pakistani colleagues can take place.
Dr. Tariq, you're muted.
So thanks for giving me an opportunity. So I would love to echo Dr. Tipu and even Dr. Aisha Moyadin, whatever they said that is actually close to my heart. So right now, I should not take that much time. I already wrote in the text that I have done so much in terms of gene functions and the pathogenic variants, and even the regenerative medicine and precision medicine that is the future of the medicine. So recently I started working at Michigan State University, but before that I worked at Children's Hospital of Philadelphia, that is second best in the United States. So there we worked on 2 different genes, 28 different novel variants, and purpose was to see which one is a pathogenic and then how we can rescue. And we did like the precision or personalized medicine, and we offered that treatment to some patients, and even we did some survival surgery, and we looked at the brain circuit malformation. So I should not talk that much in means the technical things, but the things that I would love to contribute is I would like to be the part of this forum. Even I would love to collaborate. Right now I'm working with Dr. Beg, who is actually my MPhil supervisor. He's chairman at this point of the PSF. And some of the work I'm collaborating at Aga Khan, some Qatar Foundation. So most of the time I worked actually in Korea, Canada, and United States, but I remain connected with Pakistan because I love that. That's in everybody has that feeling for the country. So I started one channel basically. It doesn't look nice to mention that, but purpose was to give the awareness to the people who are actually affected. So if they know what to do, then clinician has to do something and geneticist has to come to collaborate. So I actually release one video weekly or bi-weekly, and not that much viewership. You know that technical things are not well perceived, but I'm using different platforms. So purpose is to give the awareness and to connect the people. So hopefully we can do that, and we should, because we are the most affected people. We have more genetic disorders, and Dr. Jawad mentioned really good. He gives actually a really very good picture of what we have. And if we look at the data, we are the second most affected. And I worked on thalassemia when I started actually working on research. So I screened 400 families. So the problem in our community is that I think it's everywhere. We just do research just for our purpose. We are so selfish. I did 400 families. We never approached back those families that you have this mutation, you should keep the record, and you should not be tested again and again. There should be a data bank. There should be preventive programs connected to that. But anyhow, we were so selfish. We did, we published, we moved on, and we started new disorders, started sending samples to Europe and America. Again, I agree with Dr. Aisha, but we have some issues. We don't have that much resources, but I think I should not take that much time. I'm saying too many things, not well connected. So I should 100% echo Dr. Professor Tipu. He is the most inspiring person to me, and he has contributed a lot. So, but I would love to contribute. I have that all technical expertise that right now there is in the United States, from CRISPR to precision medicine to regenerative medicine. So all is published. So actually I'm not that much well-connected person. That's why maybe most of the people, they don't know me. But anyhow, I love to serve my country in either role.
Thank you, Professor Tariq. Absolutely. I think these platforms like, APPNA Merit and PINS and these groups and these webinars are one of the purposes to connect and bring those, young researchers like you. You have accomplished already a lot, but your energy and your, love for Pakistan would be a great asset. Next, I will invite from Pakistan, Professor Arslan Ahmed. He is the professor of neurology at Shifa, Islamabad, Pakistan. He is also the past president of Pakistan Society of Neurology and a great supporter and friend of neurology community, APPNA Merit, PINS in America, and always we look forward to his advice, and we would love to have his comments about what is going on and what is his comments for the way forward in this.
I think this was a great seminar. This has been long awaited actually, and Dr. Tipu visited here a couple of years back, and this is exactly what he was saying, that the clinicians should team up with the researchers. The problem, as Jawad also mentioned, was that we don't have time. That's the major problem. Too many patients to see, no protected time for research. So what you end up doing is only those clinicians who are interested or who run into diseases, rare diseases, or those who see the rare diseases and have the resources or the connections with the researchers, we are able to convince our patients to go for the genetic research, maybe to help them in genetic counseling to prevent the disease in future generations. So what I've been doing over the last 25 years or so, since I spent some time in Professor Tipu's lab way back in '94. That's where I got interested in genetics, and at that time we used to send our samples to Professor Tipu at Northwestern. But later on, coming back, then we developed initially associations with Jawad and Aisha, who were initially working at Shifa. It was we were able to collaborate at that time, but again, we didn't have enough resources, and then they moved to NUST and then NUMS now. These days, we are able to send some of our genetic work abroad via Jawad. At times, we try to do as much as possible in Pakistan. We are in touch with Dr. Saqib as well. Sometimes if it can all be done in Pakistan, we try to do it. And at other times, we have to send our, the DNA abroad for whole genome or whole exome sequences. But that takes time, and we are basically relying on the resources of, our foreign collaborators, and that takes a lot of time. So the way forward, I would say, is better collaboration and creating this awareness, especially as Dr. Tipu said that amongst the Gulf countries, they have been able to nearly eradicate thalassemia and these disorders just because of awareness and the resolution of their governments to prevent the disease. So that will be difficult in our scenario, but if we are able to create this awareness, it may be possible to do it. Thank you very much, Danish, for this Zoom seminar. Very, very good.
Thank you, Professor Aslan. Next, I would like to invite Chair of APPNA Merit's Subcommittee on Research, Professor Mustafa Siddiqi. He is the Professor of Neurology and Movement Disorder in Wake Forest University in North Carolina. So Mustafa, please go ahead.
It was a great discussion, and Professor Jawad, thank you for your talk. Professor Tipu Siddiqui को सुनना जो होता है वो हमेशा बड़ा thought provoking experience होता है. हमेशा वो इकबाल को quote करते हैं और इंसान को बड़ा जबरदस्त motivate करते हैं, and he always calls a spade a spade. So I always listen to what he says very carefully. So I always think about whenever there is a discussion taking place about what are the actionable items and how can we make something out of all this discussion and all these great minds who are contributing with their thoughts. So one of the things which I want to actually propose to all the participants, especially, and that has to come from Pakistan, is, I mean, what is the low-lying fruit? So I'll give you a little bit of an experience, and what I'm really getting at this is to move towards a registry for genetic diseases, and then there are issues about funding, who will fund it, and all that. But really, वो वाली बात होती है कि first deserve and then desire वाली बात होती है कि a registry can be started by a number of heavy hitters in Pakistan by joint collaboration and there are things like RedCap. I don't know if you all are familiar with it. RedCap is a free registry database available from Vanderbilt University. और मैंने जब उसको map पे देखा था, so there are about 6 institutions in Pakistan who already have a license for RedCap. So do read about RedCap. The second thing, what as I was listening to everyone, was there is a NIH national database for rare diseases. It is called RADAR, RADAR, and they have actually given a whole sort of blueprint or the guidelines how to make a registry. So one of the things which can come out of this talk is that some people in Pakistan can take an initiative regarding making a registry and really pooling these resources from different AFIP ka naam mention hua hai, then Khan has that, and then there are other institutions who have that. The good thing about genetic registries is that is hardcore data. I'm on the steering committee of some Parkinson's registries in America, और वो हमारा वाला data जो होता है वो तो सारा clinical data होता है, and that's not hardcore. There are so many factors which influence that. So genetic registry would have some hardcore data, and really papers can come out of that, careers can come out of that, or it becomes like a win-win situation for everyone. So something to think about, and that's all what I would like to comment. Thank you.
Thank you, Professor Mustafa. Now, I would like to invite a great supporter and participant of all these webinars that we are doing, director in DRAP, which is Drug Regulatory Authority of Pakistan, Professor Ubaidullah, to give his perspective from regulation standpoint or otherwise as a great at the helm of research and regulations in Pakistan. Professor Ubaidullah, please go ahead.
Thank you very much, Dr. Shahid. In fact, I will not go into details because very beautifully articulated by Professor Tipu, international scenario and the local scenario by the Dr. Saqib Mahmood as well. But important point from the national perspective and domestic scenario is the establishment of an interface or platform where researchers and clinicians can work together, because it is the very basic missing link in our country, not only in the field of genetics or genomics, rather in every field of medical sciences or pharmaceutical sciences. So my first point is that we should think for the establishment of some very interactive platform where clinician and researcher can discuss their practical problems for further discussion. Another very important from the genetic point of view is a lack of resources for the diagnosis of genetic diseases and therapies as well. In fact, Pakistan is far behind in such diagnosis, and when we are not able to diagnose, then the talking of gene therapy is a very far cry, and probably we are not in such position right now to discuss all these things. The important point for all these things is the curriculum upgradation, because I know the curriculum, both of the pharmaceutical background and the medical background, that needs incorporation of some very basic clinical trial infrastructure, which is very lacking in right now. And we are working with various councils to incorporate the basics of trials in their curriculum. In my opinion, if we would be able to proceed in this direction, then we can talk about something about the genetic gene therapy and trials, because we can see that Pakistan, we are in very initial phase of clinical trials, as already shared by Dr. Sakir Mahmood. Some companies are coming to Pakistan for their trials of their vaccines, phase 3 trial. And probably it will take much time to have trials of gene therapy and genetic engineering and genomics as well. But as a regulatory authority, DRAP, I'm pleased to share that DRAP and NUMS both have signed an MOU about 20 days back for the scientific collaboration. And this forum, I want to invite Professor Aisha and Jawad, that if any type of help you would like to have from DRAP regarding scientific collaboration or some funding from our R&D fund as well, then we would like to have to help and coordinate and move further. Your director, Orik, and your vice chancellor is very well in the picture, and hopefully we can move further in this direction. Thank you.
Thank you, Dr.
Thank you, Professor Abdullah. I apologize that we have, running out of extended time also, but very quickly, I kept our friend, Dr. Uzair from Canada at the last, because he really can give us an overview and his sense of the direction. But before that, I would invite Professor Sarwat, please quickly, in 1 to 2 minutes, give your opinion and comments, and then we will close the session part. And then if Danish has some questions from the audience, I don't know if we have 5 or 10 more minutes for that.
Yes, sir. Thank you for the opportunity, sir. I just wanted to quickly say that we kind of little bit danced around the dinner about talking about the same thing of lack of resources in Pakistan and lack of this and lack of that. I just wanted to point out that the Pakistani doctors in America have a combined income equal to many small countries. And then I think in addition to talking about the solutions, why don't we think about putting our money where mouth is and do something substantive that can be shows results in the short term and in the future? Because I really don't think that government of Pakistan, despite its all good efforts, is in a financial position at this time to invest in all the labs and all the resources that we require to bring forward all the aspiration that we talked about. Thank you very much.
Thank you. Always unique approach, and I agree, Professor Sarwat, that transfer of technology may need some investment from the diaspora, whether in healthcare or otherwise, for uplift of things that are needed in Pakistan. Last but not the least, Professor Uzair, who has really been great support for us, gathering these illustrious people and exploring the landscape of healthcare innovation as well as research in Pakistan. He is the director of R&D, the Canada-Pakistan R&D Society in Canada. Professor Uzair, please go ahead.
Assalamu alaikum ji. My just very brief 2 cents. Basically, that remind me, I met Professor Aisha once I first visited the KRL lab in G9 to meet the Qasim Mehdi, because I was helping them to find out some of the supplies from the Germany. So that was my first exposure, and then I was really privy what they were doing. That remind me we are seeing each other, say, like after 20, 25 years. Much has changed. Walaikum salam. So very briefly, I bring my 2 cents is I'm an action-oriented person, and my background is not only in medical sciences, but from the management perspective, how to make the thing happen. So that is the situation analysis what we are really discussing. So ideally, we need to apply the management principle to get the job done. As simple as that. So to begin with, we need to have a very, very brief paper. What is the scenario back in Pakistan? And what is, required to take it to the next level. So I will be really supporting for the articulation of the policy, which is from the government level, the public sector, be it the registry, with the new restructuring of National Institute of Health Pakistan. So we need to collaborate. And my request would be that we need to have a very, very brief 2-pager, but after every session, that is the landscape SWOT analysis, action point, what is required to make it happen. And I will try my best really to do whatever I can. And I'm sure that we can create a difference and a complete picture, what we have been discussing. So time is running short. So thank you to all of you. And it was really a pleasure to be in this session.
Thank you. The panelists, great comments, great thought-provoking way forward directions. Danish, do we have time for, and do you have some questions? And I see some raised hands. How should we go about it? If you have collected some important question, we can ask speakers and the panelists about them. Next 5 minutes.
Yeah, I think we can spend a little more time in discussion because it seems very fruitful. Let's not kill it before we achieve something out of it. We probably won't have time to go through one by one questions, but I think there's still some discussion points that we should highlight. I see some raised hand, Professor Saqib Mahmood, Professor Tipu. I would love to hear what they have to say. Let me just quickly add what I'm hearing, jaise Dr. Uzair ne kaha, I'm also very action oriented and I'm trying to figure out what kind of things we can put together as a proposal or a working idea for collaboration. So, it seems like there are 3-4 different aspects that are coming out, which can probably be put under a single umbrella. And that umbrella could be a human genetics network in Pakistan, and that can cover the registry for patients. We also need a database of genetic researchers. We probably need to build a database of people interested in genetic patients. So for example, there is a network in US which is called Undiseased Diagnosis Network or Undiagnosable Disease Network, UDN. And I've sent actually a patient to them that I had who I couldn't diagnose, and they eventually were able to find out the exact genetic syndrome that patient had. And so it's pooling of resources in some places, because like Professor Aslan said, not everybody will be able to genetic therapies and genetic diagnosis. हर एक के पास ये टाइम नहीं होगा कि वो ये ढूंढे कि कहाँ से कौन सा टेस्ट कराना है, कहाँ से कौन सा टेस्ट कराना है. Similarly, we need probably a database of what genetic test can be done where. एंड एक ये अक्सर सवाल मैं देखता हूँ फोरम्स पे लोगों ने डाला होता है कि मेरे पास सस्पेक्टेड हंटिंगटन्स है या सस्पेक्टेड ये है, वेयर कैन आई गेट द जेनेटिक टेस्ट डन, एंड वी डोंट हैव अ डेटाबेस. और इसी तरह अमेरिका में बहुत सी यूनिवर्सिटी की लैब्स होती हैं, जैसे प्रोफेसर साकिब की लैब है, प्रोफेसर डॉक्टर जवाद की लैब है, हु प्रोबेबली हैव सम कैपेसिटी टू डू सर्टेन टाइप्स ऑफ डिजीज एनालिसिस इन रिसर्च सेटिंग. And in US, those labs then offer that ability. If you allow us to save the sample and use it for research, we'll offer you a free report on that genetic test. And sometimes even hum Germany mein bhej dete hain ki that lab in Germany is offering, because they're doing a research project on these diseases like epilepsy, autoimmune epilepsies. If we send a sample to them, they will use it for research, but they will also send us a clinical report that we can share with the patient. And we probably need a database for that. Genetic course ki baat ki Professor Saqib Mahmood ne, I think that's very important. We need to entice and familiarize physicians about the importance of genetic studies, we need to probably create some form of a basic genetic course for physicians in general, and then we can create some extra module for example, neurology ki community ke liye genetics or cardiology ke liye alada hogi, and so on and so forth. And then of course we need a place for interactions, जैसे बात हुई पहले कि physicians or geneticists or researchers आपस में interact कर सके, whether in form of conferences, webinars or a group play. So अगर आप ये 5-6 different elements को जोड़ लें, I think that can come under a large umbrella of a genetic research network or a human genetic network or something, which have these arms and are trying to put all of these things together in forms of registries and databases and networks and courses and education and awareness and so on and so forth. And we can all probably chip in and put in an effort, including money. Dr. Sarwat, I second you, and I'll be happy to donate not only my time and expertise, but also my money for the worthy cause. Thank you. Shahid, if we're going to take comments from Professor Sakim and Professor Tipu, you can go ahead.
Professor Tipu, did you have comments and concluding remarks, sir?
Right. So just to recap, when parents look at their children, they say we can either spend this money on ourselves, buy a new car or a house, or we can spend it on our children's education. And I think every generation is faced with that. My propensity would be that that we think about the future generations. It is very difficult to correct something that has gone so bad in genetics for so long. It's very difficult. Cousin marriages, if you just look at cousin marriages, they don't produce any extra burden of disease. It's only in those people where the disease already runs as a mutation that you have a problem. So what is the long course? The long course is that we take all fertile individuals and we do whole genome sequencing on them. And we do those elements that are repeated, which cannot be covered by this kind of sequencing. That is a cost that is far less on the burden on society than that is mated with local-based genetic counseling and treatment. The physicians in this country, in the United States, don't know genetics. They have no idea. Even those who have specialized, they have no idea about genetics, or very little. They have sort of, idea based upon what they read in the newspapers. They don't have time here either. And the corporate medicine will not give them any time except to see patients and more of them. So it's a very difficult situation for the profession of medicine. In America, they are not doctors, they are providers. It's almost like people standing on the streets to sell their bodies. So you don't want that. You don't want that in Pakistan. You want to maintain your profession that has survived for thousands of years. So this, when we saw in COVID that America and the Western countries, especially America, completely failed because of its terrible medical system in spite of the waste in it. So we have to think in different ways as to how we can get the biggest bang for the buck. And I think the biggest bang for the buck is what I mentioned earlier, so I won't repeat it, is by education. If you look at all the great movements in history, they're all based upon education. And so one has to say, I want to postpone my immediate desire. I've got good quality frontal lobes, and I'm going to let this happen to the next generation and the generations after that, so that something useful comes through it. And that, I mean, all these things that you want to do are great. They're immediate and they can be done right away. And in that sense, I don't want to trivialize them. I think they're very important. They take human effort. But really, to have a vision, one has to sort of separate yourself from the present and say what is possible in the future. Now, I'm sure that there are engineers in Pakistan who can make those lasers for sequencing. And all the sequencing equipment that is needed could be made. It will give people, young people, projects to do. And first time they may not succeed, but eventually they will. If you can certainly make the nuclear bomb, you can make lasers for sequences. It depends where you want to put the effort. You'll buy machinery from America or Japan or other places. It's going to be very expensive, and then they can shut it off whenever they want. So I think related to this effort is an opportunity for, technical universities and engineering universities to come forward and say, with the clinicians and geneticists, that this is how we can help to make homegrown instruments. And I just want to digress for a minute and show and tell you about this man, Asif Sharif. I don't know how many of you know about that in Pakistan, but he has a completely new system for agriculture, which does not use fertilizers, which does not use pesticides, which needs very little water. It's a natural system, and it is very rapidly catching on amongst the small farmers and even in East Punjab. So I follow his WhatsApp thing, and it's remarkable that it is 20, 30 times more. What I'm trying to say is that he has made all the machinery himself. So now the FAO, the agriculture organization here, is calling it with a different name, but they don't have the machinery. So that machinery is all homemade. So I think it is doable. If we are determined to do it, it is doable. But we have to find people who are interested in doing it, who have the expertise, whom you want to tell what you want. And I think that's a more desirable and worth somebody's time. The rest of the stuff is already there. It just needs to be implemented. I don't want to trivialize it, but I think that the real change has to come from the bottom up, and changing our curriculum in the medical school and colleges. We are not even prepared to be good physicians. We go after 12th grade into medical school, and we have no idea about biochemistry, microbiology, the huge, giant knowledge gap that is in our physicians, because they are not taught those things. They're not taught statistics. They're not taught mathematics. So there is that other problem. But I think it can be remedied by intense courses that can be put out with modern methods. But we got to start people to think scientifically right from childhood. That's my plea, because you want to leave this a better world for your children and grandchildren, rather than try to spend millions of dollars for gene therapy for somebody who's ill now. We just have to take a deep breath and suck it in and say, what a terrible tragedy, but we don't want this to go on.
Thank you, Professor Tipu. Lastly, our speaker, before that, Professor Saqib, you have raised hand. If you can go ahead and give your comment.
Yes, thank you. Thank you. It was just a comment in the sense that there was a lot of talk about expertise, international collaboration, resources. I believe there are a very robust international collaboration in Pakistan on genetic disorders. Even now, clinicians are directly collaborating with the international collaborators. At least they are getting the diagnosis for their patients. But the important thing is what is required is national level collaboration, because we have the expertise, we have the technology, and we only need to pull up our resources need to be pulled up to at least for the genetic diagnosis. Pakistan has about 6 to 7, I believe they have NGS, but none of them is functional. Every university has a Sanger sequence, but very few are working as such. The reason being, everybody is working in isolation. Nobody is working with collaboration with each other. So if we pull up our resources, we pull up our expertise, we are able to provide genetic diagnostic services to the community in Pakistan. That's just I wanted to say. Thank you.
Thank you. Professor Jawad, did you have any last comment, concluding remarks? Professor Jawad, if not, then Danish, do we have any questions left, or we should conclude now?
It's been almost 2 hours. I think we can conclude. There is still more questions, but I think we'll have to conclude.
I think there is no other word left. Many things already said. So thank you very much, everyone, from my side. And I think it was a very excellent opportunity for me. And it was nice talk, and it's very nice to listen. So many people. Thank you very much, APPNA, Merit, and everyone.
Thank you, Professor Jawad, and our illustrious panelists, particularly Professor Siddiqi and Professor Aisha and all others. who collaborated so well in this webinar, and because of the interest. And thank you, audience, who stuck till the end. Thank you. We'll see you next week.
And I think, Shahid, we should make maybe a merit genetics group and gather people so we can continue these conversations and not drop the ball.
Absolutely.
All right.
Thank you.
Everyone.
Thank you, everybody. Thank you, Danish.
Thank you,
Shahid.
Thank you, everyone.
Thank you, Jawad.